What plays the bigger role in human development, genetics or environment? This philosophical debate has been ongoing for decades and has no clear-cut answer. Those who have studied the issue carefully agree that certain aspects of an individual are influenced more heavily by one than the other. Personality, ethics, and values are primarily dependent on environment. Unfortunately, the extended environment outside the protective sphere of parents and home often plays the dominant role in influencing who and what young people become. On the other hand, no environmental influence can impact hereditary conditions like hemophilia, Down syndrome, or colorblindness. Studies of adopted children are revealing that a certain degree of bonding occurs while an unborn fetus is still being carried in the uterus. How this may impact some of the special problems that adopted children face remains unclear.
Hi, I'm Mike Shively. Welcome to Introduction to Anatomy and Physiology. We've just completed a couple of sessions where we talked about the male reproductive system and the female reproductive system. And I want to talk today about human development. But before we get into that, do you have any questions about previous sessions? Susan?
I was wondering about the term breech birth and what that entails.
What does breech birth mean? Basically, when a baby comes through the birth canal in a normal presentation, it's called, the head passes through the birth canal first, followed, of course, then by the shoulders and thorax, and then the rest of the child comes through. In what we call a breech birth, something other than the head passes through first, or presents itself to pass through first. Quite commonly, and there's several different versions, depending on what particular part of the body is. The classic breech is the rear end trying to make it through there first. And of course, when you think about a child, you have to fold this kid kind of in half. If the buttocks presented first, stop and think, you have to fold this in half, and of course that's going to make a larger wad of neonate / fetus, neonate, coming through the birth canal. And so quite often when abnormal presentations are made, by a fetus, the OBGYN, attending physician, will attempt to turn the youngster around so that it's, can pass more easily through the birth canal. Any other questions? Adam?
So in the days before the home pregnancy test, how could they, determine if you were pregnant or not?
Yeah, you're talking about the old rabbit days, I bet. And as a matter of fact, we talked in the previous session about the hormone that causes the color change in home pregnancy tests being chorionic gonadotropin, which spills over, of course, into the urine, and then will interact with chemicals in that home pregnancy test to cause the color change. Well, the same hormone, of course, was produced before and spilled over in the urine before they had home pregnancy tests, and they used to, years ago, use female rabbits and inject urine from a prospective mother into the female rabbit. It caused changes in the female reproductive system. I don't recall exactly what those changes were, but it was chorionic gonadotropin that was interacting with the tissues of the rabbit that actually made change. Anything else that we could clear up? I want to write a word here on the overhead camera. Ontogeny, and another word, phylogeny. Ontogeny refers to the developmental history of an individual, and phylogeny refers to the developmental history of a group of animals. Basically, it comes from the same word that phylum comes from. Individual and group of animals. And it's kind of interesting, when we're talking about the biological or developmental history of an individual, all of us have exactly the same ontogeny. Now, we may have different personal histories, but our biological history is exactly the same for each one of us. And in fact, it's the same general biological history for any mammal. all mammals have essentially the same ontogeny. Now, animals that belong to different phyla will have, different phylogenies because different phyla have some peculiar developmental characteristics. But your and my ontogeny is the same, and it's the same as that of our pet dog or pet cat or any other mammal that you might want to mention. So I want to talk about the major stages of ontogeny, but let's make sure we've got this word ontogeny down. We'll go to a graphic here. Let's make sure we write that down, the biological history of an individual. That's what we mean by ontogeny, because this word itself is not in a lot of our working vocabularies, perhaps. Well, what are the major stages of ontogeny? Let's go to a graphic that summarizes those major stages. Now, this is not necessarily every stage of ontogeny, but this would be a listing in the order that they occur, and some of these we're already familiar with, and several of the others we're going to define and talk about for some time today. Gametogenesis, we've already actually talked about. In males, what do we call this? Adam?
Spermatogenesis.
Spermatogenesis. And in females, gametogenesis is called what? Susan?
Oogenesis.
Oogenesis. So we've already talked about that. We've diagrammed it in previous sessions. This is the formation of male sex cells and of female sex cells. So the general umbrella term for either sex is gametogenesis, and for males that is called spermatogenesis, for females oogenesis. Ovulation we've talked about. Now, ovulation is actually the bursting forth of a mature or vesicular follicle through the surface of the ovary. It has to go through both the tunica albuginea of the ovary as well as the covering visceral peritoneum. And remember, it's accompanied by a few of those follicular cells when it makes that passage through. So we've talked about that. Copulation, or sexual intercourse, we define that as the procreative act wherein a man's erect penis is inserted into the vagina of a woman. When the man ejaculates semen into the vagina, then the sperm within that semen can be carried on up through the cervical passageway into the uterus and up into the uterine tube, where fertilization may or may not occur. If there is a ovum passing down a left or right uterine tube at that point in time, then fertilization might in fact occur. If there's no ovum there, obviously fertilization will not occur. Incidentally, what happens to those sperm? Those sperm that get deposited in there and there's no ovum, what happens to them? Susan?
Aren't they just reabsorbed into the body?
Yeah, they die. Some of them make it all the way up through the uterine tubes and out the end of the open end of the infundibulum and get out into the peritoneal cavity, but they only last about a day or so under ideal conditions outside the male reproductive tract. So they die and basically are not heard from again. So that's the fate there. Now the next stage on this graphic is cleavage. I want to go ahead and define these terms, and then we're going to talk about some of them individually. So cleavage is actually a term that refers to the first several cell divisions of the zygote right after fertilization. Now remember, the term zygote means fertilized ovum. So once a sperm has penetrated the ovum and fertilized that ovum, then that complete cell is now called a zygote. Remember, it has 23 pairs of chromosomes. Sperm brought in 23 chromosomes. The ovum had 23 chromosomes. And together, that makes 23 pairs, or 46 chromosomes. The first several cell divisions, we're not going to say exactly how many, but the first several cell divisions are developmentally referred to as the process of cleavage. Nidation is a fancy term that refers to implantation. And this is when the conceptus, now quite a few cells in size, actually implants or embeds into the uterine endometrium. So implantation is the more common and more familiar form of what we're talking about with nidation. Gastrulation refers to the division of the early conceptus into three distinct layers of cells, what embryologists call the three primary germ layers. I think we have mentioned those a long time ago in one of our earlier sessions, but we'll talk about those some more today. The names of those three layers are ectoderm, mesoderm, and endoderm. Those are the three primary germ layers. At any rate, this term refers to the separation of early embryonic tissues into those three primary germ layers. Organogenesis, up here, refers to the interaction of the early tissues with each other to form various organs. And this actually goes on for quite some time. And the period that we call growth, or enlargement and refinement of those various organ primordia, occurs for some time during gestation. And then this is the pivotal point when we're born, birth. Or if your mother was a duck, this is when you hatch, okay? You know, coming through a different channel there. So this is pivotal point. We all understand this. Maturation refers to every part of human development between the stage that we call birth and when we reach older, quote, geezerhood. Now there's no clear-cut period, you know, when you actually become a senescent individual. My children and some of my grandchildren think that I've been there for a number of years, you know, but I don't exactly feel like I'm quite there yet, although I certainly look in the mirror and say, you know, I'm not the young man that I once was. And of course, basically most of us are someplace in here to here, and once you get to these stages of ontogeny, there's not just a whole lot to look forward to, as you can see, because there's only one more stage and it's not a lot of fun. So these are the major developmental stages that we're talking about, and I want to back off from that now and spend a little bit of time talking about them individually. Human developmental individuals often group some of these stages into, you know, in various ways. We'll take everything that happens before birth, and we'll call that the prenatal period. And everything from birth to death is often called the postnatal period. So that kind of takes all those different stages of ontogeny and groups them into two big groups: before you're born and after you're born. Quite often the period of gestation is divided up. Let's go to the overhead camera for just a second. And what they'll do is they'll say, well, let's take this prenatal period before birth and let's divide it up into, well, we'll use the one stage that we already had, gametogenesis, formation of sex cells. And then we'll talk about the pre-embryonic period. And this is basically about 2 weeks long, okay? First 2 weeks after fertilization. So that's going to include fertilization, it's going to include cleavage, it's going to include implantation or nidation, and gastrulation will occur. All of those stages that we listed separately a while ago will happen in those first 2 weeks, the ones that I just mentioned. And then after the pre-embryonic period, we've got the embryonic period. And this one lasts about the next 6 weeks. So when I put 6 weeks down here, it's the next 6 weeks. So this conceptus is now basically 8 weeks old by the time we reach the age of the embryonic period. And then basically the rest of the prenatal period is sometimes called the fetal period. I think I spelled that wrong, didn't I? There we go. When a conceptus is recognizably, in our case, human, then we quit calling it an embryo and we start calling it a fetus. So there's not a, it's not all that clear cut. Basically, based on the time periods that we have here, it's any part of gestation after the 8th week when you get right down to it. So those are ways that we can divide up the period of time before birth and stack various of those onogenic stages into different parts of this. Now, what I want to begin with today is, we've already talked about fertilization. I want to spend some time today talking about cleavage, starting in with cleavage. We've already talked about the other, fertilization, gametogenesis, and so forth. So we want to begin with this zygote. If we go to the overhead camera, we've got a fertilized ovum here, and remember that is called a zygote once fertilization has occurred. The zygote exists for about 24 hours, and within about 24 hours it divides. And when it divides to form 2 cells. Now this is mitosis, regular cell division. A little less than 24 hours after this initial division, each of these, or this will divide into 2, and this will divide into 2, so we end up then with 4 cells. And developmentally, we call this the 2-cell stage and this the 4-cell stage, and then of course this continues to happen since the initial divisions occur in synchrony. Next we'd have 8 cells, and then 16, and then 32, and then 64. After several of these kinds of synchronous divisions, we end up with a solid ball of cells where there's just too many cells to conveniently count. There's a bunch of them in there, it looks like a cluster of grapes, and it's not very easy to actually count them. When we get here, we call this a morula. This is before you can remember. All right? After these first initial several cell divisions, some of the cells start dividing at different times than others, so the synchrony is actually lost, but still there is more or less a doubling and a doubling and a doubling of the total numbers of cells. Now, the morula continues development, and with a few more cell divisions actually hollows out to form a hollow ball of cells that is called a blastocyst or a blastula. You'll see it both ways in the literature. We'll write blastocyst and then we'll put blastula down here. Barely got room. Right at the very bottom. This stage is what actually implants. And the total period of time from fertilization around to this is about a week, maybe 6 days, but roughly a week, short side of a week perhaps. This is where we are when implantation or nidation occurs. Now, the blastocyst or blastula actually has 2 clearly separate parts. This group of cells that are kind of inside here, right in here, that's called the embryoblast or inner cell mass. I'm going to write it up here. And that inner cell mass actually develops into the individual. So that's the group of cells that actually becomes you. The rest of these cells that kind of form the rest of the ball around here, around this hollow liquid-filled center, these cells collectively are called the trophoblast. And many of those cells contribute to what are called the extra embryonic membranes. So I wanted to show you, we get around here to a solid ball of cells, morula, just a little bit more development. We get a hollow ball of cells called a blastula or blastocyst. That is what actually sticks to the uterine endometrium. Now, we're a week down the road from fertilization, and there's been no source of nutrition for this conceptus yet. Doesn't receive any real serious nutrition until implantation occurs. If we go back to that overhead camera one more time, I want you to notice that we had a big cell here, smaller cells, smaller cells. Every time division occurs, the cells get smaller and smaller. Why? Because without a source of nutrition, what's happening is these cells at regular mitosis or regular cell division are simply divvying up the existing cytoplasm and organelles. And so the cells have to get smaller and smaller and smaller until we finally get down here. Once implantation occurs, now that's not necessarily going to be the case, because now there's going to be a source of nutrition coming in. But that's basically what happens for the first week of development. So it's that hollow ball of cells called a blastula or blastocyst that actually implants. Now, right after implantation occurs, right after it sticks and physically adheres to the uterine wall, which incidentally, kind of interestingly, is most typically right in the middle of the posterior part of the uterine wall. That's a very common place for nidation to occur, although it can occur in other parts of the uterine wall as well. But right after implantation occurs, the inner cell mass, or that embryoblast that we talked about that's going to become the definitive individual, actually develops into two layers, an upper layer and a lower layer, and then finally a third layer develops in between, so we have eventually an outer layer, a middle layer, and a lining or inner layer. That outer layer becomes the primary germ layer that we call ectoderm. We'll see these on a graphic in just a second. The middle layer develops into what we call mesoderm, and the lining layer develops into the primary germ layer that we call endoderm. Let's look at that graphic now, and I want to show you what definitive tissues are derived from each of these three primary germ layers. Again, the names of the layers are ectoderm, mesoderm, and endoderm. From ectoderm, we have nervous tissue. Do you remember when we developed the neural plate, neural tube? Remember that?
Yes.
Had the groove and plate and tube. And we said that, hey, the nervous tissue is actually derived from this outer layer of the developing embryo, the stuff that we call ectoderm. In addition to nervous tissue, there is some epithelium that develops from the outer layer. Primarily the membranous epithelium and glandular structures that are associated with the skin, and just the initial parts of the oral cavity and that sort of thing. Mesoderm, the middle layer of the three primary germ layers, gives rise to all muscle tissue. Now we're talking about smooth muscle tissue, cardiac muscle tissue, and skeletal muscle tissue. All of the connective tissues, bone tissue, blood, adipose tissue, cartilage, all of the connective tissues. And there is some epithelium that is derived from mesoderm as well. Primarily the epithelium that lines blood vessels and lymphatic vessels, as well as the epithelium associated with the serous membranes. Finally, endoderm, the innermost or lining of the three primary germ layers, gives rise primarily to some epithelium. So from this, you can see that epithelium develops from all three of the primary germ layers. Nervous tissue comes from ectoderm, and muscle tissue and the connective tissues are all derived from mesoderm. Okay? Any questions there? Now, I want to go back to the overhead camera for just a second. I want to call your attention back to, let's draw this blastocyst again very quickly. And we said that these cells out here were collectively formed what we call the trophoblast. Inner cell mass here, which becomes the definitive individual. And these guys out here are the cells that make up what's called by an embryologist the trophoblast. The trophoblast cells, and even some of the cells from the inner cell mass, right in here, are involved in the formation of the what we call the extraembryonic membranes. Now, what would extraembryonic signify? Just the word. What's that saying? Extraembryonic. Susan?
Not part of the actual embryo.
Not part of the actual embryo. In other words, this is not going to be definitive you. This is not, you know, once you're born, this is not going to be part of you anymore. But developmentally, it's part of the conceptus, the total conceptus. Let's take a look at the 4 extraembryonic membranes in a graphic here. And they are by name chorion, allantois, amnion, and yolk sac. Chorion, allantois, amnion, and yolk sac. The chorion itself, this first one, it develops from the primary germ layers ectoderm and mesoderm. And it forms the major part of the fetal portion of the placenta. Now, we haven't talked about placenta all that much, but the placenta has both a fetal part component and a maternal component. In other words, there's some endometrium from mom's uterus, and there's some mesoderm and ectoderm from the developing fetus that grow together to form the chorionic portion of the placenta. At any rate, chorion makes most of that fetal part of the placenta. The next one on the list was allantois, and the allantois actually contributes a little bit to the chorion and the placental formation, but the allantois does something else that's kind of interesting. Let's get rid of this graphic for just a minute, but leave the overhead camera on. I want to show you something here. The urinary bladder of a developing near-term fetus, I'm just going to write bladder, actually has, coming off the end of it, actually has a tubular channel that goes out the umbilical cord, and it goes all the way out to the placenta area. And that little channel in there is called the urachus, coming off the urinary bladder. At any rate, this channel goes out and connects up with the allantois portion of the extraembryonic membranes, and late in gestation, when the kidneys of the fetus actually begin producing a little bit of urine in the very last part of gestation, obviously this urinary bladder can only hold so much, and the waste products, so that they can be channeled off into an area where they can be absorbed, actually go through the urachus, through the middle of the umbilical cord, and actually out into the allantois area. And from then, from this area right here in the extraembryonic membrane, they can be reabsorbed into mom's bloodstream such that mom can deal with the fetal trash, metabolic trash. Does that make sense? So this name of this channel is called the urachus. It's within the umbilical cord. Every once in a while, we'll hear of a patent urachus. You know, when the umbilical cord is tied off, there may be still some seepage of urine from the now neonate urinary bladder into the surrounding area. In this case, going to be cut off, but you know that's basically they call it a patent urachus when they see urine leaking from the umbilical stump of the umbilical cord. The amnion forms a hydrocushion around the developing fetus, and so there's actually some amniotic fluid within that amnion, and so the fetus is basically swimming around in a little fluid reservoir, and that's so, you know, when mom bumps up against a sharp edge, you know, of a table or something like that, you don't necessarily get a permanent dent inside your head. Susan?
With the amniotic fluid, that's how they do the amniocentesis
Yes,
test.
when they're doing pulling out and looking at some cells from the developing conceptus, and they actually pull a sample of amniotic fluid. Obviously, this needle is going to pass through the anterior abdominal wall of the mother, pass through the uterine wall, and on into this amniotic sac, you know, within the lumen of the uterus. They'll pull out some of that fluid, and that fluid will contain some cells that have sloughed off and are just suspended in that amniotic fluid. Karyotyping and various analyses can then be done on those cells to check for certain possible genetic defects and so forth. In the unborn, they can check in the unborn. Now the fourth extraembryonic membrane that was on our list was called a yolk sac. And in mammals, the yolk sac is basically insignificant and vestigial. In most mammals. It's not insignificant or vestigial in a duck-billed platypus or you know, spiny anteaters, a couple of those other mammals that actually are egg layers. But in us and in most mammals, the yolk sac is a vestige. But in birds, reptiles, and amphibians, of course, it's a big deal because those animals do not have development of the conceptus within, in most cases, within mom's uterus. They're actually, in most cases, in an egg that's laid outside the mother. And so the yolk sac then serves as a source of nutrition for the developing conceptus in that manner, in an external manner. Now there are some reptiles, you know, some snakes give birth to living young and so forth, but even those in particular reptiles typically have eggs that just are retained within the body and still have a significant yolk sac. Anyway, those are the four extra embryonic membranes. Let's see, I started out telling you the primary germ layers that were involved in each. In the chorion, it's ectoderm and mesoderm. In the amnion, it's ectoderm and mesoderm. In the other two extra embryonic membranes, it's mesoderm and endoderm. So the two primary germ layers that are involved in allantois and yolk sac are mesoderm and endoderm. When a woman is pregnant, we commonly divide that gestational period up into 3 periods that are called trimesters, basically roughly first 3 months, second 3 months, last 3 months. And we often say that the first trimester is really quite critical. The reason for that is in those first 3 months, the cells are differentiating into the parts they're going to become, and any impact on that process will have far-reaching consequences from a developmental standpoint. We're not here trying to suggest that the second trimester or the third trimester are not important, But clearly, it can be shown from an experimental and a philosophical point of view that the first trimester is really quite critical because of all the various differentiations that cells must go through to decide whether this group is going to become liver, whether this group is going to become 5 fingers or 6 fingers or 2 fingers stuck together in a syndactylous relationship, and so forth. I want to put up a graphic here that, and I don't want you to memorize the times and what's going on here, but I think it's an interesting graphic on significant developmental events. If we say day 0 is when fertilization occurs, then by the time we're 4 or 5 days down the road post-fertilization, then we get to that blastocyst stage that actually implants or nidates. So, 6 days or so is when the actual blastocyst sticks to the uterine endometrium. It's sometime during the 2nd week after fertilization that gastrulation occurs, the formation of those 3 primary germ layers. The heart forms sometime during week 4, and it's beating, with almost right away, right after it forms. So 5th week or so, the heart's beginning to beat. You can usually detect it by 6 weeks or so. Fingers and toes separate out here at about 6 weeks of age. This is post-fertilization now, 6 weeks post-fertilization. And we're 3 months down the road by the time the fetus is 50 grams. Now, in case you don't remember, anybody tell me how many grams there are in a pound about?
Kilogram is 2.2 pounds.
So we're not real good at conversions right now, huh?
Another
Well,
500 grams.
it's 450 or so.
3.59.
Yeah, about 450. So we're talking about, roughly a 1/5 of a pound is all this conceptus weighs out here at 12 weeks post-fertilization. And so quite a bit of the increase in size, of course, is going to occur, in particularly the last trimester. And finally, we get up to, the 38th week or so, then birth is getting to be imminent. Ladies that have had multiple children, quite often the first child is a little bit longer. Gestation takes a little bit longer for the first birth. We're talking a few days longer. We're not talking about weeks or anything. And then quite often subsequent births will be just a little bit shorter in total gestational length. Now, once we're born, we've got postnatal development to talk about. And let's take a look at a graphic that summarizes the major stages of postnatal development. The first period, postnatally, is called the neonatal period. This basically includes the first 30 days after birth. For all practical purposes, we're talking 1 month. After that, we say that this child is an infant. And infancy encompasses the period of time from about 30 days up to 2 years. 30 days after birth to 2 years. 2 years was a time period that was arbitrarily selected to divide what we call infancy from childhood. So a 2-year one, a child that's, a day short of 2 years of age is still considered to be an infant, even though they're going to be running around typically. And then after they have their second birthday, they're considered officially by developmental folks to be a child. And childhood lasts an indefinite period of time from one individual to the next. It's basically from 2 years clear up to what we call puberty or sexual maturity. And sexual maturity is happening right in here is what's going to divide childhood from adolescence. Now, you know that sexual maturity happens at different ages for different individuals. Basically, a person is considered to be sexually mature when they began producing gametes. So for boys, that's going to be when spermatogenesis commences. And for girls, it's going to be, when ovarian cycling begins. Okay? That makes sense? Now, adolescence goes for, well, who knows how long. My wife says I'm still there, but my hair color would say otherwise, actually. Adolescence goes basically from puberty up to this indefinite stage that we call adulthood. So basically, you can think of adolescence as kind of encompassing the teenage years, This is a difficult period of time for a lot of young people. I saw a bumper sticker once that said, hire a teenager before they forget everything, because teenagers kind of think they know a whole lot, and of course they do. In reality, they're not nearly as experienced, though, as those of us who have been around a few more years. That's advice that they don't often take well, in fact. And those of you who are parents, you know what I'm talking about. And those of you who will become parents, listen up, because it starts out bad and it tends to get a little bit worse sometimes. Children can be the most wonderful thing and the biggest headache, that you ever encounter. Migraine waiting to happen. After adulthood, we've got a couple other stages. Senescence, that we briefly talked about before, and finally death. And that's the end of life as we know it. Now, if we go back and think about those stages of ontogeny that we listed at the beginning, we started out with the first stage that we had up there was gametogenesis. And then once we reach sexual maturity and become an adult, adolescent/adult, then we are, as an individual, are undergoing gametogenesis is occurring in our bodies as well. So basically, the great circle of life that Simba was Mufasa got across to Simba, was the fact that, it is one big circle, starting with gametogenesis, not our own, but gametogenesis occurring in our parents, right? And then we get to the gametogenesis stage, and then we can procreate, we can produce children. And then we get old and die, but our children can reach the gametogenesis stage and begin to produce their own children. Hopefully we live long enough to see that happening. One of the greatest joys that I have personally had, of course, is watching one of our children in a public place deal with their children, our grandchildren, throwing a real temper tantrum right in the middle of the Kmart floor. You know what I mean? Mm-hmm. And then I'm thinking back, I remember, Dory, when you did that, or I remember, Jeff, when you did that, and how concerned I was at that time. But as a grandparent, you just accept it. Hey, that's going to happen, and it's not nearly as embarrassing and difficult to deal with, particularly when they're the ones that have to take them home, and you get to enjoy them for a while as a grandparent. But at any rate, that's kind of human development in a nutshell. Now I blasted through it from top to bottom, and I didn't give you much opportunity to interact with me or to ask any questions. What kinds of questions do you have about anything in regard to human development? Yes, Susan?
What are the types of things that they can determine by the embryonic fluid? What's that composed of? And
Amniotic fluid?
Yeah, you said cells
Yeah.
that have sloughed off, but I'm...
Of course, the majority of it is water.
Okay.
And there are amniotic cells that come from the lining of that membranous sac that slough off into it. And of course, those cells, being originally from the zygote, through several cell divisions, will have the exact same genetic material within it. So they can actually karyotype or actually do, those are chromosome preps,
Okay.
so they can actually spread these cells out on a microscope slide if they're caught in the right stage of mitosis. Spread them out and actually examine for certain chromosomal aberrations. We haven't actually talked about chromosomal aberrations, but you know the normal human has 23 pairs or 46 chromosomes, but in some cases individuals have trisomies, or 3 chromosomes where they're only supposed to have a pair. In other cases, individuals have a single chromosome where they're only, a monosomy where they're supposed to have a pair. Probably the best known and most commonly talked about trisomy is trisomy 21, which is commonly called Down syndrome, or sometimes it used to be called Mongolism. I think they're getting away from that name because of the stigma associated with it, but Down syndrome was a common trisomy. Anyway, some of those kinds of things can be determined from amniocentesis, the preparations that are made from the fluid that's collected during amniocentesis.
Can
Now,
they tell the blood type and everything
Um,
for the RH factor bababies, or is that
you know,
developed with the different
I'm
diagnostic?
not sure, Susan. Now, amniocentesis is not routinely done. I mean,
Right.
We're only going to do this if we have suspicion that a certain kind of thing might be the case, and we want to get either a heads up on what may happen and what kind of immediate neonatal care this new individual might have, or in some cases, the parents may elect not to bring this particular individual into the world because of, if it has this particular problem, it's not going to have a quality life based on the health care that's going to be required to maintain its life. So, and those are certainly tough decisions, and I don't fault anybody for the decision that they might make in regard to some of those issues. Was there a question over here, Adam?
Yeah, what age does or what length of time does a fetus become viable? When can it live outside the mother?
Well, that's very interesting. In the last few decades, there have been tremendous inroads made in neonatology and so forth, to the place where, premature babies can actually be significantly more premature today than they could, say, 30 years ago. I don't know, I can't tell you what the youngest one has been, but it's amazingly few months. Now, these little fellas and gals come out so tiny. Our last 2 children, Jeff was 9 pounds, 10.5 ounces, okay? He was full-term baby. He came out basically kind of walking and talking and looking for trouble, all right? Sherry was our last child. She was 4 pounds and 14 ounces. She was, basically slightly less than half of Jeff's size when she was born. And I think I mentioned to some of you before, she was so small that we actually bought a case of preemie diapers and went to Kmart and bought doll clothes to put her in, because the smallest infant clothes were just too big for her. She'd get lost in there. Now, 4 pounds and 14 ounces is a really small baby. By the same token, that's not anywhere close. There have been babies that have been so premature that they weighed in the vicinity of a pound that have made it, in fact, with the neonatology to the place where it is today, and they're just able to do so many more things with these preemie youngsters than they were able to accomplish before. So that's a good question. I don't know the answer, but it's a good question.
While we're talking about recent developments, there's a lot of controversy about embryonic stem cell research. Where do the embryonic stem cells come from?
Well, they can actually collect some of those from some of the offal or the placental tissues that are passed, which are going to be discarded or thrown away. There's some cells that are still living in those tissues. That is certainly one source. And it's interesting, isn't it? I, being the biological scientist type, I'm not opposed to those kinds of procedures because it's intriguing what they can do, and they may be able to use some of those cells in ways that could, in fact, perhaps save a life or improve lives in certain disease syndromes. But by the same token, I will be the first to admit, with the advances that have occurred in biomedical science, we are now getting to the stage where we are able to prolong life and save individuals that 40 years ago would have naturally not, they wouldn't have survived. And as we save those individuals with advances in biomedical science, at that same time, we are preserving the genetic pool that gave rise to them. In other words, the human race is no longer surviving on the natural selection criterion. We are now, because of the aids provided by biomedical science, we are in an unnatural selection, and we are actually weakening, if you will, if you allow that word to be used, we're weakening the human race with the advances that are occurring in biomedical science, because we're allowing individuals, I shouldn't say a word allow, that kind of sounds like we're making the choice, but we're preserving the genetic pools of individuals, and those individuals are becoming adults in some cases and procreating, such that those weaker gene pools are being preserved. We can take some parallel examples, say from, let's go to the dog racing world, okay? Racing greyhounds, selected for what? Speed. Selected for what goes along with speed? Musculoskeletal soundness. These dogs, if they are not fast on the track, you know what the owners of those racing dogs do?
Sterilize them or put them down.
Sterilize them, make them into pets, or put them to sleep. So the racing dog industry actually selects for musculoskeletal soundness. A racing greyhound as a breed, they don't have any canine hip dysplasia because they're extremely sound. They've got musculoskeletal health that's unbelievable. On the other hand, Boston Terriers have such big heads that without veterinarians around at the time of birth, that breed would have become extinct a few Boston Terrier generations ago, simply because they're, I don't know if you're familiar with Boston Terriers, they're cute little fellows. They look like they got a tuxedo on all the time, And their heads are so big, though, in the puppy stage that they quite often can't pass through the birth canal of the mother dog naturally, and so they have to be born cesarean, with some veterinary assistance. And so that's just an example, from the animal world of what we're doing to preserve. The breeds of dogs, we started out a few hundred years ago. We had, this is the veterinarian coming out in me, please allow me. We had, just a few breeds, and we used those dogs for various things. Then dogs became kind of more of a companion animal, and I own one of those. I have a Maltese, He's one of those little dogs that looks like if you put a handle on him, you could mop up the floor, a little Moppet type dog, but he's cute and he's lovable. On the other hand, some of the breeds that we've developed just as companions, like Shar-Peis, that have the skin folds, they have a lot of skin problems because of the moisture that gets down in those folds and so forth. We developed the breed, why? Because we thought it was cute. We preserved that gene pool by giving it official breed status and, breeding and selling these particular puppies. I'm not trying to pull exact parallels between that and what we were talking about just a second ago, but it's the same kind of thing. We're preserving certain genetic pools, and medical science is doing that. As a human race, we're not as hardy, is what I'm saying, as we were a few generations ago, simply because we have all kinds of assistance. When I was a kid, I'm sure I had a bunch of earaches and so forth, but I probably have taken in fewer antibiotics in my life than you folks have taken in, because you know you're a generation or so removed from me, and now it seems like every time a child gets sick we run down and get the antibiotic, and as a consequence, what are we doing with all those antimicrobials? We are developing some very, very resistant strains of bacteria to the place where we now have Staphylococcus organisms that there's only 1 or 2 antibiotics that'll touch that particular strain, and with, you know, next year or within a couple of years that strain will develop a resistance to that new antibiotic, and we stay about, you know, just barely ahead of the resistance development that's occurring. So we as a race are becoming weaker. Any other interesting questions that we could raise? I didn't mean to wax so philosophical on that, but it is an interesting question and does deserve a little bit of time. Well, let me tell you what we have coming up in the next couple of sessions. We have actually only a couple of sessions to go, and we've actually postponed the integumentary system purposefully until this period of time. So I want you to read the chapter in your textbook regarding integumentary system. That's going to be involving the structures that we call skin, hair, and nails. So we'll spend some time on that on the next session. And for our final session, I think we're going to spend some time doing what I call a regional review. And it'll be kind of a lot of interaction back and forth between you and me, involving some of the regional aspects of anatomy. And I want to talk about some of the kinds of things that'll summarize and pull together some systemic anatomy with some regional anatomy, and some things that we haven't talked about as far as regions of the body. We talked about the big regions, I think, in one of the first or second sessions that we had, but I want to go into some other areas and talk about quadrants of the abdomen and the 9 official regions of the abdomen and some regions up here in the cervical area. So that'll be what we talk about in the next 2 sessions.